From the RSNA, welcome to the Radiology Imaging Cancer Journal podcast hosted by the Trainee Editorial Board. I am Yashbir Singh from the Mayo Clinic. We wanted to highlight the ongoing special collections we have. The neuroradiology head and neck collection will be concluding soon, and we will be launching the pancreatic hepatobiliary one next. Dr. Srinivasan is editor in chief for Neuroradiology 1, and Lassie and Pallavi are leading pancreatic hepatobiliary collection with help from Dr. Luke on the interventional side. Today, we will continue exploring this year RSNA theme, imaging the individual, with our special guest, Dr. Lacey McNally. Dr. Lacey McNally is a professor of surgery and Stephenson Chair at the Stephenson Cancer Center, where she serves as a co leader of the Cancer Therapeutic Program. She is internationally recognized for pioneering work in developing contrast agents and nanovehicles for cancer detection, monitoring, and treatment. Dr. McAnally conducted the first clinical trial using multispectral optoacoustic tomography in the United States and currently leads three phase 1 clinical trials. Her research target multiple cancer types including breast, pancreatic, ovarian, and hematological cancers, as well as inflammatory disease such as colitis. Dr. McAnally serves in Women's Leadership Council for the World Molecular Imaging Congress and contribute to the advancing the field both internationally and nationally. She has a long history as an NIH study section reviewer, including her previous experience as a standing member of NIH imaging probes and contrast agent study section, and in the academic industrial partnership mechanism. She also serves as a deputy editor of Radiology, Imaging Cancer, and section editor for Molecular Imaging and Biology, shaping the direction of molecular imaging research and clinical application. Dr. McNally, as a researcher working in Department of Surgery, you occupy a unique space between basic science and clinical medicine. How has this interdisciplinary environment shaped your research on contrast agent and molecular imaging?
Well, thank you for having me on your podcast. Number one, I initially went to veterinary school, and so I've always been kind of clinically focused within my laboratory. In fact, for our lab, the most important thing is to actually move things, not just have success in mice, but to move things into the clinic. And in the last couple of years, we've been able to do that. And we've run several clinical trials now off of stuff we've developed and off of things that we have interacted with. So for our lab in particular, it's actually a symbiotic relationship and quite important.
So, building on that translational perspective, you conducted the first MSOT clinical trial in the United States and are currently running three phase 1 trials. What were the biggest challenges in moving this imaging technology from the lab to human patients, and what has surprised you most about its potential across cancer types and inflammatory disease like colitis?
As a basic researcher, our lab is really accustomed to writing more traditional manuscripts that are published in art in journals such as Radiology Imaging Cancer. We are also very accustomed to writing NIH grant applications. However, writing applications for the FDA was our biggest hurdle. It was something we did not have a lot of expertise in when we first started. And FDA applications, particularly in our case, an IDE or investigational device exemption, was a very different mechanism. It was very different from both manuscripts and from grants, and it required a lot of extra advice and help from other people, as well as from consultants from our own IRB department, many Q-sub pre-IDE discussions with the FDA. When we first started with the FDA was not accustomed to our device. And so there had not been a device tested when we first initially started this. The FDA had questions too. They were interested to see if we were using radioactivity, which we weren't, but they initially couldn't fathom how we could actually image tumors using hemoglobin without radio labeling it. So it was a long journey for both myself and the FDA to actually get our first trial up and going. We had a lot of clinical collaborators that were very excited about the opportunity to non-invasively detect cancer immediately prior to surgery. So there were quite a number of challenges that we had starting out, but now that we have actually done this successfully, like most other things, once you've had success, it's easier to do it the second time than it is to do it the first time. So we wouldn't have been able to be successful without help from our clinical colleagues for sure. So I think that's the first thing, and now that we've been able to be successful with that, that's actually one of the reasons I moved to the University of Oklahoma, because they're well known for being very high in phase 1 clinical trials, particularly for cancer, and so that was the reason I moved here in the first place. It was very helpful to have administrative people that could assist us with communication and making sure we were on time. I would think that's probably about it.
So, your clinical trial experience brings valuable perspective to your editorial role. As a deputy editor of Radiology Imaging Cancer, you review countless manuscripts. From your editorial seat, what separates a good imaging research paper from a truly exceptional one? Are there like common pitfalls that you see that researchers should avoid?
My experience on the editorial board would say the most common pitfall that at least that I see in manuscripts is a lack of focus. A lot of times authors will think that it's most important to put in every piece of information that they have that possibly relates to any topic at all that they're writing about. But it actually is a detriment to their paper because it diffuses the meaning and the outcomes of their paper and the importance of their paper. It distracts the reader, it gives a lot of tangents, and so the readers get lost. The editors get lost too. So I would say a focused paper with a lot of depth in particular is the most important thing. And looking at a particular, having multiple readers on your study, don't just have one, have multiple. Have multiple assessments of a similar topic so that you get more accuracy in your data. Have more subjects. We're looking to try to make sure that the outcome of the paper holds true to the data, holds true to the text. And unfortunately, a lot of times that's not the case, where the paper will say one thing in text and the data suggests something completely else in the manuscript. And that's not a very good outcome for the manuscript. So I think more focus in the paper, look at something in a deep scale instead of lots of small things very superficially. Have a focus, have a goal, and have a particular path that you're traveling for the paper that makes sense in a direction. Don't take a lot of tangents and a lot of, oh, this could also be exciting. Oh, this also could be exciting. Try to stay on target.
So let's dive deeper into manuscript quality. Imaging research lives and dies by its figure. What makes a compelling publication worthy figure in molecular imaging? Could you just share specific guidance on how researchers should present their imaging data to tell the clear scientific story?
Well, the first thing you have to do is you need to have enough resolution on your image. If your image is quite pixelated, that makes it very difficult for us to look at. That's the first thing. Second thing, have scale bars. That means both size bars and dynamic range bars of your contrast that you're looking at. In my personal opinion, it's better to have physical values that you're having on those dynamic contrast bars as opposed to just saying it's between 1 and 0 or between 1 and 100 where it's normalized. It's easier to see the difference in a particular number. And the reason for that is as a reviewer or an editor, you can evaluate that and compare that to your control. So if your control has a top number of zero point 0.1 and your experimental one has a top number of zero point 0.2, then that may not be a very big difference. It could be, depending upon what you're looking at, or it could not be. But it makes it a lot easier for the reviewer and for the editor to know exactly what your data is, at least in my opinion, when it's not normalized. That would be the first thing. Second thing, I would make sure you have different aspects of your image to be annotated. Having arrows or stars or other information that shows what you're trying to look at or anatomy is also tremendously beneficial. If you send an image in and it has no annotation and you're expecting the reviewer to just be able to read your mind and know what your image is supposed to be, that is not necessarily the recipe for success. The other thing I would recommend too is it's also nice to show a control. No matter what your control is, you should have one. And it's easier for a reviewer or an editor or a reader be able to assess your data if it shows either before and after, or a control and a treated, or disease not diseased, anything like that that you can show a comparison of what your data is, I think is helpful.
Beyond figures, the peer review process itself can make or break a manuscript. Having served as both an editor and reviewer, you have seen this process very closely from both sides. What advice would you give to the researcher when responding to the reviewer comments, and how can they revise their imaging manuscript to not just survive but thrive in the review process?
The first thing I would do is I would actually read the comments, and then I would not respond to those comments immediately. I would come back maybe the next day or the day after and then read the comments again with fresh eyes. That's the first thing I would do, because no matter what, everybody generally thinks that something that the reviewer said was either incorrect or they didn't agree with or whatever. But usually, if you think about it a little bit more, the reviewer's comment makes sense, at least meaning that perhaps you didn't make something clear and the reviewer potentially misread something that you wrote in a different way, or they interpreted something differently. So the first thing I would do is whenever you get your comments back from a manuscript, read them and then don't respond immediately. Let them wait. So that's the first thing. Second thing, you need to respond to every single comment. If you don't like one or you disagree with it, you need to respond to that with justification, including several different references that can back your reason why you disagree with the reviewer. It's also very important to try to respond with extra data or extra references to every single comment. It is challenging on the editorial side if the author says, I disagree with this comment for every single comment. That's not usually the recipe for success to get your paper accepted. That usually says to the reviewer that you are completely in disagreement and not receptive to anything any of the reviewers said. So I would strongly disagree with doing that. Although we have seen people to do that. So, if you're one of those people, maybe it's a way to improve your review response is to agree with the reviewer at least somewhat. The second thing I would do is it's easier if you will have a comment and you're going to write what the reviewer comment was first, and then exactly as it's written in your letter that you get from the editor, and then respond immediately below that. That could be, see line 11, we have adjusted this text and included these extra references, and include the references. Don't just say that you included the references, actually do it. And even in the response document, I would write what references you included in that comment. So it's immediately below. So it's easy for the reviewer to check off and say, yes, this person actually followed through. The other thing I would really recommend doing is, if there's a comment, or a lot of times, like I said, we see images that are just not annotated. So just on the front end, check that your images are annotated. Just be easy on the front end. It's a requirement. You need to do it. If they're not annotated, please do not assume that the reviewer does not have any idea what your image is on, or that the reviewer just doesn't know what your field is, or the reviewer is incompetent, or whatever else poor thing you can say about the reviewer, and just do it. If your paper is on glioma, maybe somebody is going to look at your paper and their specialty is colorectal. They may not be able to identify all parts of the brain. You don't know. So I think extra annotation is always helpful. The reviewers spend quite a lot of time going through manuscripts and trying very hard to do a good job. We don't send manuscripts out to people just randomly. They're selected for expertise in your field. They're also, maybe it's not your field in colorectal surgery, maybe they're experts on the type of imaging modality they've used. Maybe they're experts in statistics. Maybe they're experts in mouse imaging and you're doing human imaging. But there's always room for learning. And as the author, it is your job to make sure that you are appropriately conveying the data in your paper. It's your job to make sure that you are being clear and concise and focused. And any comments that you get from the reviewer are specifically to improve your paper or to tell you what is unclear or to how to improve your paper. Nobody is ever writing comments that are mean or that are rude or that are trying to get at somebody in a vindictive way or any of those terrible things. It's all trying to help your paper to be the best it can be.
As someone who wears multiple hats, leading a research program, serving on a journal editorial boards, and reviewing a major publication, you have a unique 360-degree view of scientific publishing. What advice would you like to share with the scientific community about conducting and publishing quality research?
Having a theme here. You need to be focused. Focus is the theme and have quality controls. I think having well controlled research is really the key for success. That means having maybe multiple control subjects. It may be having multiple control scenarios where maybe you're having a control for your contrast agent and a control for your disease and a control for your imaging modality. It's also important to, if you're doing, particularly for cancer, if you have something that is a MRI image that you're proposing in your manuscript, it's nice to have a histology image to back up that cancer was actually found in that particular patient that you're showing the image of. Thing that's really important is try to have multiple assays or multiple parameters that you're using to try to show a similar outcome. Also is very beneficial to ask a statistician to review your data and not just use Excel on a t-test and to use proper statistics. So I can't stress enough how important I think it is to actually consult a statistician before you submit your manuscript instead of after you submit your manuscript. I think that's another key.
Finally, any message that you want to share to all the trainee editorial board member?
There's a real opportunity, especially on our trainee editorial board, because on our trainee editorial board, our trainee editors get to have the opportunity to actively participate and see how the review process is in real life. It's not mock, it's every week we have a meeting, and I think it gives them a huge opportunity to learn. We've had a lot of trainees that have graduated out of our trainee editorial board and have learned so much that they are now full associate editors in our journal and others because of the amount of information that they've been able to learn about the process and how important it is to respond to reviewers succinctly and focused and how best to do that and what particular aspects of a manuscript make it a good manuscript or make it a manuscript that's acceptable and that will be cited. So I think there's a lot of opportunities. I think the major thing that you want to do is you want to put your hat in the ring, so to speak. You want to actually apply. Don't assume because you are a trainee that you may not know something or may not have sufficient expertise, or maybe you're from a small school, or maybe you have English as a second language, or anything. The most important thing is to try and put yourself out there and give yourself an opportunity to learn.
Thanks for spending time with us today. Special thanks to our guest, Dr. Lacey McNally, for her valuable insight. If you are enjoying the conversation, please rate, review, and subscribe wherever you are listening. Your interactions help more listeners find the show. Check out Radiology Imaging Cancer. Link to the journal will be in the show notes. Today, you heard from our team, and special thanks to our editor, Kelly Myers. We will see you next time on Radiology Imaging Cancer Podcast.